Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on the topic of variation and evolution, and in particular on genetic engineering. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started.
Hello and welcome to this tutorial on genetic engineering. So today's tutorial is quite a chunky tutorial. As you can see, we have quite a few learning objectives. So we're going to be going through the process of genetic engineering, applications of genetic engineering, some negatives of genetic engineering, and also we're going to be going into more detail of the actual mechanism behind it. And we have quite a few specification points, and the specification is quite detailed on this, so perhaps sit back and enjoy the video for now and come back to these at the end and just make sure that you know what's going on and what you need to understand. So our first specification point of today. So genetic engineering involves introducing a gene from one organism into the genome of another organism. Okay, so simple as we're engineering the genes of another organism by introducing a gene from one organism into this organism. So genetic engineering is also known as genetic modification. So you might hear it as these two different terms, but they mean the same thing. And so genetic modification or engineering is a very topical discussion. Now there's lots of controversy around this because many people believe that we shouldn't be messing around with our DNA. However, others believe that it is a necessary tool for the future of science. So we're going to discuss some of these points later on in the tutorial. So now let's talk through the actual process of genetic engineering step by step. So genes can be cut out from the chromosomes of humans and stuck into other organisms. So for example, we can genetically engineer bacteria to make them produce insulin, so that we can mass produce insulin in a fermenter. So we select a desired phenotype, that's our first stage. So we've got to identify the desired characteristic or phenotype that we need. And in this case, if we want to make insulin, the desired characteristic is insulin production. So we know our pancreatic cells are very important in producing insulin. So we might go to the pancreatic cell, for example. So here we would find the insulin gene. So we'll find this in all cells, but in the pancreatic cell it's going to be most active and probably most accessible to scientists too. Whereas in other cells, this insulin gene might be turned off. So we go to the pancreas cell, we find the insulin gene in humans, we call the humans the donor organism because they're donating this insulin gene, and this gene will be responsible for insulin production. So we then take this gene, we remove the insulin gene from the human cell and insert it into the bacterium. So our human cell is our donor organism. Our bacterium is our recipient organism because it's receiving the insulin gene from the human. Now the bacteria are now described as genetically modified, and we often use a vector for this step, which we will learn about later on in this tutorial. So you can see that all we're doing is we're inserting our human insulin gene into our recipient organism, which is our bacteria in this case. We're cutting the gene using these enzymes, kind of like scissor enzymes, so they act as molecular scissors. They're called restriction enzymes. And we then isolate this to be added to the bacterium DNA. So as I said, we find the bacterial DNA, so there's either a loop of DNA in the bacteria or plasmid. We're going to be using a plasmid in this case. Again, we cut it using restriction enzymes. We insert our little human insulin-producing DNA into the plasmid, and we then form this recombinant DNA. So all we've done is we've cut out a section of the bacterial plasmid, we've inserted our insulin gene, and together we call this our recombinant DNA because it's the recombined plasmid with the human insulin DNA. And remember, the plasmid is bacterial DNA. So as I said, this is our genetically modified bacterium. We then culture the recipient. So we have our insulin gene in our bacteria. What now? Well, we can get the bacteria to replicate. We do this using a fermenter, a large-scale fermenter. So our bacteria replicates. As it replicates, it's going to produce more and more identical copies of itself, therefore allowing mass production of insulin-producing bacteria. Simple as, okay? So, genetic engineering in plants. So, genetic engineering of crops can produce genetically modified crops. So, I'm sure you've all heard of GM crops. So, farmers can use genetic engineering to produce better crops. So, this will give them more money and help the consumer. They might produce crops that are resistant to disease or have large fruits. And crops can also be made that are resistant to herbicides, which means that herbicide can be used to kill the weeds, but the actual crop itself survives, therefore allowing for maintenance of the crop yield. Now note that in the previous tutorial we had a look at selective breeding, and you can see how it's different to genetic modification, because in selective breeding we're selecting the crops that are resistant to disease, for example. So this statement here, in selective breeding we're selecting the crops that are naturally resistant to disease or that naturally have large fruits. But in genetic engineering we are actually combining genes so that we ourselves make crops that are resistant to disease or have large fruits. So you can see the difference here. So crops can also be genetically engineered to aid humanitarian situations. So for example, golden rice was produced to be given in regions that suffered from vitamin A deficiencies. Now, golden rice contains a substance called beta-carotene that provides its golden colour. And this supplements the vitamin A in vitamin A deficient people and therefore aided with vision problems across the world. However, due to debate over GM crops, golden rice was not produced worldwide, but instead it was given to regions that actually suffered from the vitamin A deficiency. So you can see how the debate behind genetic engineering is kind of holding back us as humans moving forwards with the concept of genetic engineering. Now, genetic engineering in animals. So bacteria can be genetically modified to produce insulin, as we've seen. But in the past, we used to use insulin from animals. But this was actually found to be inefficient. So therefore, bacteria have been engineered instead to produce insulin. So insulin is used to treat diabetes, which is a condition that leads to an inability to control glucose levels in the body. And that's why producing insulin in this way, using insulin-producing bacteria put into a fermenter, is really, really useful because this insulin can be harvested and injected to treat these patients. Okay, so that's why we find using bacteria that have been engineered to produce insulin absolutely amazing. So medical research also uses genetic engineering. So after the advent of using bacteria to produce insulin, there are many more avenues that are being considered through genetic modification. So for example, we can use genetic modification to treat HIV, sickle cell anemia, Huntington's and cystic fibrosis. So lots of genetic diseases may actually have the potential to be cured or treated in this way. So we've talked about the positives of genetic engineering, so let's have a look at some negatives. So to summarise, we have health risks, ethical hazards and unknowns. So I'll go through these now. So health risks associated with genetic engineering. So some GM foods can lead to health risks. So for example, they could contain chemicals that cause allergy or toxins. Ethical wise, so some do not believe that it is ethically fair to modify organisms and make newer life forms. Maybe due to religious beliefs, for example. So they might believe that they are playing God by doing this. So it might not be ethically acceptable. Also, there are unknowns. So the concept of GM is very new, very novel. So we don't actually know the long-term effects of genetic engineering. And also people worry that they could lead to a huge effect on the ecology of organisms. which could in turn lead to negative effects on the populations of wildflowers and organisms. And we don't know all of the effects of eating GM crops because, as I said, it's a very novel concept. So there could be unforeseen consequences that we only find out in the future. Now make sure you learn these advantages and disadvantages because they could well come up in your exam, and there'll be a really simple few marks if you do know them. Okie dokie, so that's specification points covered on this slide. So now there's another set of specification points because your exam board seems to love genetic engineering. So for the higher tier, it is necessary to describe the steps of genetic engineering. So to do this, we've already gone through the basic steps, but we're just going to go through it in a little more detail. So as we know, genetic engineering involves removing DNA from the genome of one organism, which is the donor organism, and placing it into the genome of another, which is the recipient organism. So how do we cut our required gene? Well, we use these enzymes that act as scissors. Now, these enzymes are called restriction endonucleases, so learn this term because you will really impress your examiners if you use them. So we cut our DNA using restriction enzymes, restriction endonucleases. This allows us to cut the genome of the human cell and remove our insulin gene. So we're using an insulin example again, but remember this could be any advantageous gene that we want to cut out of the human genome. This insulin production gene is then isolated to then be added to bacterium DNA. So we use a vector. So our vector can be either a virus or a bacterial plasmid. So in our first slide we saw how we can use a plasmid, and we'll use a plasmid again because this is the most common method of doing it. So in this case we would insert the insulin gene into a plasmid vector. And ligase enzymes are used to stick the insulin gene and plasmid together correctly. So we have our plasmid isolated from our bacterium. We cut it using restriction endonucleases again. We use the same restriction endonuclease. That's really important for you to remember. So the same restriction endonuclease that we used to cut the human DNA to extract the insulin-producing gene, we use the same restriction endonuclease to cut our bacteria, and that's just because it cuts it in a unique pattern which then allows both these ends to be joined together if they've both been cut by the same pair of molecular scissors. And we then join these two using our DNA ligase at sticky ends. So the way I like to remember this is ligase, so it sounds like the word ligate, right? And ligate, to ligate, means that we stick together two things. So in this case, we're sticking together our human insulin-producing DNA and our plasmid, so we're using DNA ligase. So we then have our vector with our insulin gene. We call this our recombinant DNA because it has been recombined. And we then introduce it back into our donor organism, so into our bacteria. We then call this genetically modified bacterium because it contains recombinant DNA. So our plasmids are transferred back into our bacterial cell, and then we can introduce it into a fermenter where the bacteria are going to multiply, and we're going to get lots of insulin-producing bacteria. So just quickly going back to the enzymes that we used. So remember we had our restriction enzymes, which were our restriction endonucleases, and we had our DNA ligase enzymes. Our restriction endonucleases we used to cut our DNA, both our human DNA to extract our insulin-producing gene, and also we used it to cut our bacterial plasmid to make space to insert the human insulin gene. We also used DNA ligase enzymes, which were our sticky together enzymes. We used that to make our recombinant DNA, and our DNA ligase did that by joining up our plasmid to our human insulin gene. Now, both sets of enzymes are specific to certain base sequences, okay? So remember that. So now that we've covered all of the theory behind genetic engineering, we're going to have a look at a practice question. So as per usual, I'll read through the statements, I'll pause the video, you will pause the video and go through and decide which are true or false, and I'll go through them once you unpause the video. I'll go through the correct answers. So statement A, one potential disadvantage of genetic engineering is unknown future consequences. B, genetic engineering is a form of selective breeding. C, genetic engineering requires a plant vector. And D, golden rice has been a success worldwide. So pause the video here and have an attempt. Okie dokie, so let's go through some answers. So A is absolutely correct. So this is a potential disadvantage of genetic engineering. It's just simply that we haven't had it around long enough to actually know what its long-term consequences will be like. B, genetic engineering is a form of selective breeding. No, we've already established that selective breeding is just seeing which characteristics that you like and taking those organisms and breeding them together, whereas genetic engineering is actually recombining DNA using a donor organism and a recipient organism. C, genetic engineering requires a plant vector. No, as we've seen, we can use bacteria and viruses, so not only plant vectors. D, golden rice has been a success worldwide. No, as we saw on the slide about golden rice and vitamin A deficiency, it wasn't introduced worldwide because of the potential controversy of using genetic engineering. So it was just introduced into regions that had this vitamin A deficiency. Okay, brilliant. So well done for today. Lots and lots of specification points, so do go through them and just make sure that you've understood everything. And I'll see you for the next session.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.